Hello everyone, I am Sarita Rawat, Assistant Professor, School of Pharmacy, Suresh Gyan Vihar University, Jaipur, Rajasthan. Today is our next lecture of Pharmaceutical Microbiology. Today's our lecture topic is Pharmaceutical Spoilage. So, learning objective of today's lecture is to study about pharmaceutical spoilage, to study types of pharmaceutical spoilage, to study about assessment of microbial contamination and spoilage. Now, learning outcome of today's lecture included you are able to know about the pharmaceutical spoilage, type of pharmaceutical spoilage, assessment of microbial contamination and its spoilage. So, before starting the lecture, let's start with the definition of spoilage. So, spoilage of a pharmaceutical product and drugs are referred as the changes in a physical and chemical properties in such a way that the formulation of therapeutic agent gets deteriorate and become not stable for use. Now, microbial spoilage includes the contamination of pharmaceutical product with the microbes and which leads to spoilage of the product that affecting drug safety and quality and it's not intended for use. Now, it is shortly defined as deteriorations of pharmaceutical product by any microbial contaminants is known as spoilage. So there are several factors that cause spoilage of the pharmaceutical product, like physical factor. Physical factor included deteriorations of pharmaceutical factors like heat, temperature, evaluation, and others. In chemical factors, deteriorations of pharmaceutical product because of chemical reactions like oxidation, reduction, hydrolysis, proteolysis, ionization, etc. Now, talking about the microbial contamination or microbial spoilage of the pharmaceutical product, so it can be happen by the deterioration of pharmaceuticals due to the contamination of any microbial cell like bacteria, fungi, molds, etc. Now, why to study about spoilage? Why we need to study about spoilage? So when we see, we have to study about spoilage to determine the microbial contaminants present in any formulation to avoid health hazard related to the microbial infections, to acquire knowledge about source and different type of microbial contaminants that leads to different doses and their deteriorations action. Now, to avoid the financial problem for the manufacturer due to the loss of product, and to find out the limits of microbial content in the pharmaceutical product, prenatal, oral, and topical preparation. Parental, oral, and topical preparation that cause infections after microbial contamination, and serious conditions can be seen in the cases of typhoid fever by the injection of contaminated thyroid tablet. So you can see we have to determine the spoilage previously before coming out in the market, so it can prevent the chances of spoilage and losses to the pharmaceutical companies and also the health hazard to the patient. Now, types of microbial spoilage of the pharmaceutical includes physiochemical spoilage, chemical spoilage, and biological spoilage. So when we say about the physiochemical spoilage, so it has a variable growth, gas production, physical spoilage like olfactory and coloration factor. Now, when we see the chemical spoilage, so it is hazard, hydrolysis, acetylations, depolymerizations, and degradations of the product Then the biological spoilage, it release of the toxins, microbial metabolites. Now, physiochemical spoilage. So, in this kind of spoilage, there are some changes are caused by the microbial species, and due to these changes, the physical properties are also get altered or deteriorate. Thus, it is called physiochemical spoilage like variable growth. So microbial cell from a variable layer over the surface of the pharmaceutical formulation. This layer or the presence of the microbial cell can be clearly seen by the naked eyes. For example, layer of modes over syrup or sugar containing products like tablets, creams and ointments. Now, coloration. So, colorations occur due to the alteration in the component, chemical nature, and their changes are caused by the change in the pH of the formulation and redox of the product. So, production of some other metabolites by the microorganism. For example, Pseudomonas species, microorganism metabolite, white coloration of blue, green, brown. So, surface deteriorations of tablet containing biological product by some modes. Now, when we talk about the gas production, so some microorganism contained in the pharmaceutical formulation produces gas by their metabolic activity, and from gas bubbles, both form over the formulations. So, products containing carbohydrates or starchy material are more susceptible for the gas production. For example, the production of CO2 in the syrup that caused by osmotolerant molds and yeast like aspergillus species and the zygosaccharomyces species and calabacilla species that produce the gas in the cream and ointment containing vitamins and proteins. So, disulfovir oxide sample organic compound are also produces hydrogen sulfate in the suspension. Now, when we talk about the physical spoilage, so there are some changes in physical appearance caused by the microbial cell activities. For example, the emulsion, some microbial cell causes hydrolysis of oil phase, cause changing in the oil-water equilibrium and make emulsion unstable. So hydrolysis of oil also affect the pH of the formulation and alter the stability of the emulsions preparation. And in syrup, microbial cells metabolize sugar and molecules and due to which the concentration of the syrup changes and the product become unstable. Now, the next changes is olfactory spoilage. So, olfactory spoilage include spoilage by some microorganisms and modes generate unpleasant smell from the product. So, this kind of spoilage mainly caused by microbial cell that produce sulfur containing gas like sulfur dioxide and hydrogen sulfate. So, they also cause the fishy smell due to the formations of fatty acid along with the odor generates by the amine and alcohol production. For example, contamination in the syrup of tolu by the penicillium species that produces toluene-like smell. Now another is actinomycete produce smell of geosmin in the water phase used for formulation. Now, coming to the next type of spoilage, that is chemical spoilage. So, chemical spoilage basically occur due to the some chemical reactions in the composition. So these are the various type of chemical spoilage in the pharmaceutical compound. These occur due to a various type of chemical reactions that mediated by the contaminating microorganism like hydrolysis. So hydrolysis, some bacteria cell contain enzyme that catalyze the hydrolysis of the pharmaceutical. For example, atropine hydrolyzed by the pseudomonas bacteria, aspirin hydrolyzed by the bacillus and clostridium species. Now, acetylation. So, acetylation, some microorganism cellular enzyme caused acetylation of drug and caused loss of activity. So, for example, chloromaphenicol acetylation caused by Staphylococcus and Streptococci gram-positive bacteria by the enzyme chloromaphenicol acetyl transferase. Then the another chemical reaction is depolymerization. So depolymerization is the process in which the polymers are degraded to their monomers. So a lot of polymers are used in the formulation of many type of pharmaceutical preparation as diluents, binders, thickening agent, suspending agent, and etc. For example, starch, that is glucose, so it is depolymerized by the bacteria amylase. Pectin is galactouronic acid and it is depolymerized by the bacteria pectinase. Then dextrin, glucose, it is depolymerized by the bacteria dextrinase. Then cellulose, glucose, depolymerized by the bacteria cellulose. Now, the another chemical reaction is degradation. So degradation is due to the microbial contamination. The active therapeutic agent or the formulation's ingredient can be degraded or metabolized. For example, penicillin degraded by beta-lactamase containing bacteria cell and prednisolone degraded by the Aspergillus species. Now, the next kind of spoilage is biological spoilage. So, some bacteria cell contaminated the pharmaceutical and utilize the various compounds that are present in the formulation to their metabolite activity. So, due to these metabolic activity, the microbial cell produce certain chemicals which they release in the pharmaceutical preparation, and this is called biological spoilage. So mainly there are two type of chemical that are released by the microorganism. The first one is microbial toxin, that are several microorganism produces toxic molecules that may cause spoilage of pharmaceutical formulation, such as endotoxin that produced by some gram-positive bacteria like E. coli. So, microbial metabolite next. So, microbial metabolites, the bacteria metabolites are the biosynthetic product from microbial cell. And bacterial cell produce various metabolites which cause product spoilage because of these metabolites are toxic to humans. For example, there are different types of amines and organic acid from bacteria cell, metabolites from the fungi and moths. Fungi and moths are more specifically grown on the formulations having talc, kaolin and starch. Now, coming to the tabulation of some formulation ingredient and their agent which cause spoilage. So, the formulation, chemical compound and drug, and contaminating species and agent. So, first one we have polysaccharides, that is agar, starch and cellulose. So, it is contaminated by Bacillus, Pseudomonas and Clostridia species. Then we have protein and gelatin. So it is contaminated by Aspergillus species and Penicillium species. Then cream and emulsion. So it is contaminated by modes. Aromatic compounds contaminated by Pseudomonas species, gram-negative bacteria. Then acetyl salicylic acid, so it is contaminated by Actobacter species. Then the syrup of tolu, it is contaminated by Penicillium species. Atropine eye drop contaminated by Corneobacterium species. Then prednisolone tablet contaminated by Aspergillus species. Then hydrocortisone tablet contaminated by Clostridium herbarum. Rose hip preparation contaminated by anaerobic bacteria. So these are some examples of the formulation and their contaminating agents. Now, the factors that affect microbial spoilage. So, by understanding the influence of environmental parameters on the microorganism, it may be possible to multiple formulations to create conditions which are favorable as possible for growth and spoilage within the limitation of patient acceptability and therapeutic efficacy. So the first thing is size of the contaminant inoculum. So low level of contaminant may be present in a product, but it would cause low rate of deteriorations. Ingredients contaminated by a high level of microorganisms which cause appreciable microbial degranulations. However, inoculum size alone is not always a reliable indicator of likely spoilage potential. So, a very low level of Pseudomonas in a weakly preserved solution may suggest a greater risk than tablet. So, containing high number of fungal and bacterial spoilage. Now, the next factor is nutritional factor. So, nutritional factor, gross microbial spoilage of a pharmaceutical product generally required appreciable growth of the contaminating microorganisms within the dosing form. And most of the organic and inorganic ingredients act as potential carbon or nitrogen substance for the microbial growth. And the complexity of many formulation offers considerable nutritional variety for a wide range of microorganisms. The use of crude vegetable or animal product in a formulation provides an accidentally nutrients environment. Additionally, nutrient environment even demineralized water prepared by the good non-ion exchange method will normally contain sufficient nutrient to allow the sufficient growth of many waterborne gram-negative bacteria such as Pseudomonas species. Now, moisture content and water activity. So, microorganisms require readily accessible water in appreciable quantities for growth to occur. So by measuring a product water activity, it is possible to obtain an estimate of the proportion of uncomplexed water that is available in the formulation to support microbial growth, which is by using the formula AW water activity, vapor pressure of the formulation, vapor pressure of the water under similar conditions. So most microorganisms grow best at high water activity. Hence, formulation can be protected from microbial attack by lowering their water activity with additions of suitable level of sugars, polysaccharide, polyethylene glycol or sodium chloride or by drying. Now, the another factor is pH. So, extremes of pH prevent microbial attack. Around neutrally, bacteria spoilage is more likely with the report of Pseudomonas and related gram-negative bacteria that are growing in antacid mixture. So flavored mouthwash and in the distilled or deionized water. Our pH 8 example with soap-based emulsion spoil is rare, and in a product which are low pH stable, for example fruit juice, flavored syrup with pH of 3 to 4, mold or yeast attack is more likely. Now, yeast can be metabolized organic acid and raise the pH to level where secondary bacterial growth can occur. Now, the minimum water activity that supports growth of the microorganism. So Clostridium botulinum supports the water activity at 0.95, Bacillus cereus 0.95, Pseudomonas aeruginosa 0.95, Salmonella species 0.95, Streptococcus aureus and Candida species and Saccharomyces so 0.90, Streptococcus aureus 0.86, Penicillium species 0.82, most spoilage yeast 0.88, most spoilage mold 0.80 and osmotic yeast at 0.90. So this is the minimum water activity that support the water growth of any microorganism. Then another factor is storage temperature. So storage of the pharmaceuticals could occur potentially over the range of 20 degrees Celsius to 60. Now, the particular storage temperature may selectively determine the types of microorganisms involved in the spoilage. So, a deep freezer at minus 20 degrees Celsius or lower is used for long-term storage of some pharmaceutical raw materials and short-term spoilage of the dispensed total parenteral nutrition feed prepared in the hospital. Now, reconstituted syrup and multi-dose eye drops packed are some dispensed with the installations to store in a cold place such as domestic fridge 0 to 12 degrees Celsius, partially to reduce the risk of growth of the contaminants inadequately introduced during use. Then, conventionally, water for injection should be had at 80 degrees Celsius or above after distillation and before packaging, and the sterilization to prevent the possible regrowth of gram-negative bacteria and the release of endotoxins. Now, redox potential. So the ability of microbes to grow in an environment is influenced by its oxidation reduction balance, that is called redox potential, as they will require compatible terminal electron acceptor to permit their respiratory pathway to functions. So the redox potential, even in fairly viscous emulsions, may be quite high due to the appreciable solubility of oxygen is the most in the most fat and oils. Now next is packaging and design. So packaging can have a major influence on the microbial stability of some formulation in controlling the entry of contaminants during both storage and use. Considerable thought has gone into the design of containers to prevent the introduction of contaminant into the medicines for parenteral administration, owing to the high risk of infections by this route. Self-sealing rubber wards must be used to prevent microbial entry into the multi-dose injections container following withdrawals with a hypodermic needles, and where medicine rely on their lower water activity to prevent spoilage. Packaging such as strip foils must be of a water vapor proof and material with fully efficient seals, and cardboard outer package and labels themselves can become substrates for the microbial attack under humid conditions, and preventions are often included to reduce the risk of damage. Now, coming to the source of contamination. So, contamination can be gained entry into a production process stem from the several sources such as personals, poor poor facility design, incoming ventilations air, machinery and other equipment for the production, raw material and semi-furnished material, packaging material and utilities. So different media used in the production process as well as for cleaning and clean room clothing. So we can see in this picture the source of contamination. Basically, the microbial contamination sources included facilities, personals, utilities, water gases, materials, raw material, reusable resins and membrane filters, and then the process that involves in the productions like close and open production, then equipment, assembly, cleaning, sterilization. So these are some common source of contamination by microbial. Then, when we talking about one to one about these types, so personals who supervising or performing drug manufacturing or control can be a potential source of microbial contamination and a vector for other contaminant. The main reason for contamination for the personnel include lack of training, dirt contained between the operator's hand and starting materials, primarily packaging material and intermediate of the bulk product. Inadequate personal cleanliness, access of unauthorized personnel into the production, storage and product control area. Then inadequate gowning and personal protective equipment, and malpractices like eating food, drinking beverages, or use tobacco in the storage and processing areas. Now, building and facility. So, the building and manufacturing facilities may also contribute to the contamination. The main reason reason of the contamination due to the facility issues included, for example, insufficient size and inadequate uh, organization of the space, leading to selection error of like mix-ups, cross-contamination between consumable raw material, in-process material and finished product, inadequate faith and pest control, rough floors, walls and ceilings, lack of air filtration system, improper lighting and ventilations, poorly located vents and drains, inadequate washing, cleaning, toilet and locker facilities to allow for sanitary operations, cleaning of facilities, equipments and utensils, and personal cleanliness. So these are the building and facility by the contamination charges increased. Now talking about the equipment, so the equipment and utensils used to in processing, holding, transferring and packaging are the common source of pharmaceutical contamination. The main reason for contamination from the equipment include inappropriate design, size, material leading to corrosions and accumulations of static material and or adulterations with the lubricant, colourants, dust and still sanitizing agent, and also improper cleaning and sanitization, preventing proper cleaning and maintenance, improper calibration and irregular services, and also deliberate use of defective equipment. Now coming to the materials as a spoilage source, so the raw materials used for the production can be a potential source of contaminations. The main reason for the contamination from the raw material include storage and handling mistakes, causing mix-up for the selection error, contamination with the microorganisms or other chemicals, degradations from the exposure of expensive environmental conditions such as heat, cold, sunlight, moisture, etc., improper labeling, improper sampling and testing, and use of material that fail to meet acceptance specifications. Now, the next is manufacturing process. So there are various opportunity for the contaminations of raw material, intermediates or packaging material throughout the manufacturing process. The main reason for contamination during manufacturing process include lack of dedicated facilities to manufacture a single product, improper cleaning in between batches to minimize the amount of product changeovers, use of an open manufacturing system exposing the product to the immediate room environment, use of an open inappropriate zoning, absence of an area line clearance according to approved process following each cleaning process and between each batch, lack of cleaning status labeling on all equipment and material used within the manufacturing facility. Now, to minimize the risk of manufacturing contaminations, manufacturer product in a campaign with the appropriately qualified cleaning process and check preferred in between batches to minimize the amount of product changeovers. Utilize a closed manufacturing system. This is where the product is not exposed to the immediate room environment and vice versa. Perform an area line clearance according to approval process, following each cleaning process and between each batch campaign. Zone the facility and use the cleaning status labeling on all equipments and material used within the manufacturing facility. Then HVAC system. So HVAC system for heating, ventilating and air conditioning. A poor HVAC system can be a potential source of microbes growth and a transportation mode for dispersing contaminants throughout the manufacturing facility. The main reason of contamination is due to HVAC issues include accumulation of organic material in or near HVAC air intake, ineffective filtrations of the supply air, insufficient magnitude of pressure differentials causing flow of reversal, erroneous ratio of fresh air to residual air, inability to assess ventilations, dampers and filtrations from outside the manufacturing area and non-directional airflow within the production of primary packaging areas. Now, assessment of microbial contamination and spoilage. The first thing is physical and chemical changes. So, it is the changes of different pharmaceutical formulation include microbial contamination and spoilage, change in the velocity, pH, emulsions, stability, and loss of surface activity of formulations. Index microbial spoilage, measurement of oxygen compositions of the product, and it can also include the degree of oxidative attack and microbial growth. Now the sterility test, so testing which confirm that products are free from the presence of variable microorganisms. It claimed to be the sterile or free from the variable microorganisms. Test is conducted by the competent and experienced personnel in an adequately clean room with laminar flow cabinet facilities. All injectable and ophthalmic preparations are sterilized, hence these preparations are tested by the sterility test. Now, assessment of variable microorganisms in non-sterilized products. Non-sterilized products are tested for variable microorganisms for detection of pathogens and total variable count. The microbial terms pharmaceutical product can be divided into two groups, sterile and non-sterilized. Non-sterile drug must safely the appropriate microbial purity criteria and they are included in the pharmacopoeia monograph. So pharmacopoeial studies are prepared specifically with a view to ensuring that the medical product is therapeutically effective and safe for the patient. Now, estimation of pyrogen. So, pyrogen is a substance typically produced by a bacterium which produces fever which introduce or release into the systemic circulations. So the lipopolysaccharide and lipoproteins which compromise a major part of the cell wall or of the organism, gram positive, gram negative bacteria are called endotoxin, which are the most commonly called pyrogens. So to test the pyrogens presence, two tests are used. First is RPT test and second is LAL test. So RPT rapid pyrogen test and LAL test is limulus amoebocyte lysate test. Now, how we identify the E. coli? So, the identification of E. coli is by sport indole positive oxidase negative gram negative role. Then the fermentation of lactose observed on the EMB or MAC. So, if it is gram positive, so check hemolysis of BAP. So, if non-hemolytic, then do PYR test and negative E. coli or positive not E. coli IT by kit. But if bacteria hemolytic is E. coli, then if the test is negative, then do MUG test. So it can show the bacteria beta hemolytic E. coli, or if there is negative or positive, so in negative IT by the kit, and if there is positive, then E. coli is present. Now, identification of Staphylococcus aureus. So, gram stain, gram positive coccus, catalase with the oxidase and streptococci. So, this is all the process. Now, for the gram positive cocci, so we have catalase, then we have the Staphylococcus aureus. Then we have to perform this test. Now, we have concluded some questions regarding this lecture. You can easily answer these questions while going through the lecture. So, the question included, what are the cause of spoilage of the pharmaceutical product? What is pharmaceutical spoilage? Which bacteria can spoil the pharmaceutical product? How pharmaceutical and food products are spoiled by the microbes? What are the type of spoilage? What is milk spoilage? What are the sign of food spoilage? What are the 5 characteristics of food spoilage? What are the sign of yeast spoilage? And what is yeast spoilage activity? Now, for the reference purpose, all the lecture reference is taken from Stephen P. Diner, Norm A. Hodges, and then from the Pharmaceutics, a Science of Doses, Form and Design, second edition, and from the Theory and Practice of Industrial Pharmacy, third edition, from the Cooper and Guns Tutorial Pharmacy, from Microbiology, fifth edition, United States Pharmacopoeia, Indian Pharmacopoeia. So you can definitely check out these references for further knowledge. This is all about today's lecture. Thank you for attending today's lecture.